Welcome to the GEOTOP-A seminar. Our speaker today is Steve Levine, who will tell us about unmasking a hidden DNA supercoil relaxation activity in a site-specific recombination system. Take it away, Steve.
Thank you, DeWitt. Good morning, good afternoon, good evening, and happy Valentine's Day. Thank you for, thank the organizers for the opportunity to speak to you today. I tend these days to generally more frequently speak to audiences that are in the translational medicine crowd rather than the physical sciences crowd, but we'll see how this goes. So my laboratory is focused mainly on genome organization and the enzymes that facilitate that organization, regulate organization, and so forth. And we use a combination of experimental and computational methods to get a better understanding of enzyme mechanism and DNA physical chemistry, basically, using circular DNA as a model system. And so we've worked on DNA, aspects of DNA topology that are shown here in this slide, for example, using gel electrophoresis and atomic force microscopy to visualize topological intermediates, analyses of pathways of enzyme mechanisms that I'll talk about in more detail in the second half of the talk, and also some kinetics experiments that we've begun to marry with DNA topology to better understand some of these systems. So I think I'll start by just reviewing briefly some DNA topology. I think this is known to most of you, but it's worth just reviewing, I think, for the sake of completeness here. DNA topology basically involves supercoiling, knotting, and catenation of DNA. Catenation is not shown here because the main focus of today's talk is supercoiling and knotting. But these are natural biological properties of DNA that are essential for compacting genomes, which is a biologically very, very significant problem. In the case of the human genome, you have about 2 meters worth of DNA in every cellular nucleus, and that has to be compacted into a structure that's maybe 10 or 15 microns in diameter. So essentially the problem is taking the equivalent of taking a piece of wire that's maybe a few tenths of a millimeter in diameter and 100 miles long and compacting all of that material into an object the size of a basketball. And so how this occurs in the cell and how this kind of entanglement is managed is a key problem in biology. And we can make lots of progress at least on smaller scales by looking at circular DNA molecules and their properties as modified by various types of DNA recognizing and DNA metabolizing enzymes. So supercoiling is a structure of DNA that's observed when DNA is either overwound or underwound. And topologically, it's related to a quantity that I think everyone knows, the linking number, which is related to 2 geometric properties, the twist and the writhe of double helical DNA. Now, in addition to supercoiling, the axis of the DNA can also assume a non-trivial topology. And one example of this is knotting of the DNA. As shown here, this is an example of a positive trefoil knot, the simplest knot that one can make using a circular string. And that topological sign actually turns out to be important in what I'm going to discuss today. Each one of these crossings can be either right-handed or left-handed, and correspondingly has a mathematical sign, either positive or negative. Now, the DNA of most organisms is negatively supercoiled. And that's usually the starting point for the kinds of experiments that we do, because it's easy to isolate negatively supercoiled DNA from E. coli or other cells. And in the case of bacteria, DNA is negatively supercoiled to the extent of about one negative turn, or one turn of, excuse me, one turn where the DNA is underwound with respect to its relaxed state per 200 base pairs. So this gives us what we call the superhelical density, or specific linking difference of minus 0.05. So ratio of one negative turn per 200 base pairs. Now, the enzymes that modulate these properties, there are many categories of these enzymes. Two that we've been particularly interested in are type 2 topoisomerases, which carry out duplex passages of DNA via a double-stranded break in a DNA helix. And these can reduce or increase the linking number in steps of 2. These enzymes also can generate non-trivial knotted structures of the DNA. So in this instance here, you have a type 2 topoisomerase that's carrying out a strand passage, and it changes the linking number by plus or minus 2, but at the same time can also change the knot type from K to K prime. The system that I'm going to focus on today belongs to a class of enzymes called site-specific recombinases. And site-specific recombinases can also change the knot type, and do so by making double-stranded breaks in 2 specific DNA sequences, and then reconnecting those DNA segments in such a way that you form a chimeric or hybrid recombination site. And that can change the knot type simply by carrying out a duplex segment passage in the same way that type 2 topoisomerases do. What's less well understood is what happens to the DNA twist during those operations. So those mechanisms are reasonably well understood, with some caveats, as you'll see, in terms of what they do to knot type, but not so well understood in terms of what they do to DNA twist. And the reason why the DNA twist question remains somewhat obscure is because generally when you analyze knot type, you nick the DNA, you cause a single-stranded break in the DNA, and this is necessary for technical reasons in order to resolve the different knot types that are the products of the reaction. So all of the information about DNA supercoiling is lost and is generally ignored. As you'll see, what we do is we follow both knot type changes and also supercoiling changes over the course of an enzyme reaction. And so that information is valuable because it enables us to better understand reaction networks that these enzymes conform to. So that's going to be the main focus of the second half of the talk. Now, why do these enzymes exist at all? Today's topic is Cre site-specific recombination, and Cre is an enzyme that's encoded in a bacteriophage genome. Bacteriophages are viruses that infect bacterial cells. And the reason why Cre exists is because the phage genome is a circular DNA molecule. And when that circular DNA molecule is replicated, and this is true for any circular genome, in fact, when these molecules are replicated, there is some residual linking of the DNA circles that has to be resolved at the end of replication in order for those circular molecules then to segregate either to daughter cells or to make new virus particles. In the case of Cre, there is a pair of sites called loxP. These are 34 base pair DNA sequences that are recognized by the Cre protein, and then Cre protein carries out a set of biochemical reactions that allows for the two circles to be unlinked at the end of replication. And you can see at the bottom of the slide here a representation. By the way, the red and the blue here are the axes of the DNA. So in many of the diagrams, you'll see the double helical structure of DNA is not shown. We're looking just at the trajectory of the duplex DNA axis.
Steve,
But yes.
sorry, what does it mean? What the Cree stands for?
I used to know the answer to that. It's a lot of these gene product names evolved historically. Yeah, I'll have to
Is
get
it
back
an
to you on that.
acronym for something or?
The gene, yes, it's a gene name that
Oh, okay.
was given back before I started this kind of work. So probably in the 1980s or maybe even before that.
Thank you.
Yeah, sorry, the name escapes me right now. But it was given a name that at the time seemed appropriate, but I think it's generally lost. Okay, yeah, so the essential biological details here are that there's a pair of these sites called loxP, 34 base pair specific DNA sequences that are recognized by the Cre protein, and the Cre protein is also encoded on the bacteriophage genome, expressed in infected bacterial cells, and then that protein carries out a set of DNA transactions that ultimately lead to two unlinked circular molecules. And now those molecules can partition. And there are a number of such enzymes that belong to the same superfamily that operate in similar but not necessarily identical ways to Cre. So those enzymes constitute what we call the INT superfamily of site-specific recombinases. Now, what's going on biochemically? Well, this is a representation of two loxP sites. Cre protein is shown here as the pink oval, and two Cre protein monomers bind to each loxP site. So these loxPs now associate via protein and protein DNA interactions to form what we call a synaptic complex. The strand exchange reaction involves breakage of both DNA duplexes, so formation of two double-stranded breaks, and then by means that are still not entirely clear, exchanged in a reaction that religates the DNA and now forms a pair of chimeric loxP sites. The chimeric loxPs are indistinguishable sequence-wise from the parental loxPs. So the DNA outside those sites, however, is not identical and therefore can be used to track experimentally the progress of this reaction. So one can actually follow the kinetics of recombination using restriction enzymes and other tools. So after breakage and religation of the DNA, the sites dissociate, the protein dissociates, and now you have a pair of recombinant molecules. So this is all summarized here in this slide. Now, from a topological point of view, if you carry out this process on molecules that contain DNA supercoiling, you can actually convert those supercoils into non-trivial knot crossings. And actually, depending on whether the sites are directly oriented or inversely oriented, you can also generate catenated structures. So in this example, on the top half of the slide, you have negative supercoils converted into the crossings of a two-noded catenane or hop link. And the formation of the hybrid sites here is shown through joining the separately colored arrows and arrowheads.
Steve, I got a question. How do you know that these sites are anti-parallel aligned like this? So you can see that.
Right. I will have more to say about that toward the end. We've actually looked at the geometry of the longest lived intermediate in this reaction. So we can say something about what the geometry of alignment is, at least for the longest lived intermediate structure during synapsis. But you're absolutely right. I mean, this is a cartoon, obviously, and it's it's a two-dimensional representation. The actual geometry of the sites is probably more complicated.
Thank you.
Okay, but the absolute orientation of the DNA segments, the loxP segments along the plasmid, is critical here. If the sites are directly oriented, i.e., they point in the same direction as you follow the path of the DNA duplex, you get catenanes forming. If the sites are inversely repeated, then you get a knotted DNA product. And in this case, you go from the unknot to a positive trefoil knot. Studying the catenation reaction is a bit more difficult than the knotting reactions. So I'm going to focus today on the knotting reaction, and therefore everything that I'm going to talk about involves synthetic plasmid DNAs that have the sites in inverse orientation. Okay, so we don't have a generalized formula like White's formula for knotting and linking number changes. One thing that tends to get overlooked is that when you convert negative supercoils, the naturally supercoiled state of DNA as isolated from bacterial cells, for example, those negative supercoils actually get converted into positive knot crossings. And so if you simultaneously study both supercoiling and knotting, you need to keep track of the sign inversion that takes place. And that has important consequences in terms of things like the free energy of the DNA products, which will depend in detail on the topology. Okay, so our strategy is to investigate DNA topology altering enzymes by using circular DNAs in vitro. And the idea here is to better understand the mechanisms of those enzymes themselves. The knotting and catenation of circular DNA molecules, of course, is well known and well studied. This is not something we invented. And in fact, Nick Cozzarelli and DeWitt have spent a good portion of their careers using these kinds of approaches to understand topological enzymology. As I said, the influence of supercoiling is often not directly monitored. It's hard to study because generally the supercoiling is released when you analyze the knotted or catenated products of recombination. So what we've tried to do is to use an approach that's based on time-resolved reaction pathways to study the behavior of knotted supercoiled DNAs that are generated by Cre-loxP recombination. And this is, I want to give a shout out to this classic paper by DeWitt and Nick Cozzarelli, which very thoroughly investigated some of these systems, including the Cre system, although the main emphasis of this paper is on the bacteriophage lambda integrase system. My recollection is that this paper had a gestation period of something like 5 or 6 years. But that's easy to understand when you read the paper because it's a true knot topology mathematical tour de force. Okay, so experimentally, we work with a plasmid that has inversely repeated loxP sites. They're separated by almost 900 base pairs. Along this minor domain, there's overall, the plasmid has a length of almost 4,000 base pairs. And therefore, this experimental system, which we've used in other contexts, provides a good vehicle for trying to learn more about Cre recombination. And what we did was to take a negatively supercoiled plasmid. This a little later on, I'll show you some work involving positively supercoiled DNA. But we took the negatively supercoiled form of the plasmid and then carried out some kinetics experiments where we analyzed simultaneously knot type, recombination efficiency, or percent inversion of the plasmid, and superhelix density. And most biochemists, when they use Cre in vitro or in vivo, one thing I forgot to mention is that Cre is often used to construct transgenic animals and to carry out various kinds of gene fusion or gene excision experiments in living cells. Often very short incubations are used relative to the time courses that we use in our experiments. So often the enzyme is incubated with DNA for 30 minutes, and then the results are analyzed. We've been using protocols where we extend the incubations out to about 2 hours. And so this is what the results look like in such experiments. So these are the gel on the far left here is a knot type analysis of Cree. And this is a typical incubation that goes out to only 30 minutes. But what you see are the different DNA bands that correspond to different knot types that appear as a function of time. Remember now, all of these molecules are nicked. One thing that's notable here is that a lot of the DNA corresponds to the unknot. And that is, I think, a signature of these systems having evolved to generate mostly unlinked products. But nevertheless, you can see different knotted forms here on the gel. And we actually quantitate the formation of these different knot types over the course of the experiment. We also measure the extent of inversion, which tells us the overall progress of the reaction, and that we can do that by quantitating the amount of DNA in the parental form, these two bands here, versus the inverted form of the plasmid, which corresponds to these bands here.
Steve, I've got a question. For
Sure.
the knot type, you have a 6-noded knot, so that isn't a torus knot.
It's not, and I will come back to that.
Okay. And you've got 7-1. It looks like there's not much of it or something.
7-1 is actually there. It looks like it's cut off a little bit. That's
Okay.
the 7-1 band right there.
Gotcha.
Yeah, so these are all torus knots. And that's a characteristic of these systems which involve random synapsis of a supercoiled DNA, the so-called random collision model. Again, that's something I'll come back to when I talk a little bit more about modeling this process.
Steve,
Yes.
do all these DNAs have the same length?
Yes.
Okay, thanks.
These are so you can think of these as being not topoisomers. These are molecules that all have the same length, just different topology. But they are all nicked. So they all contain a single-stranded break, which relaxes any excess supercoiling. And that's where the information about supercoiling is lost. But we recover that by separately analyzing changes in the linking number. And now, so these are so-called topoisomer gels. These are gels that are run in the presence of an intercalating drug that enables us to resolve DNA molecules by linking number. And what you can see is that over the course of the experiment, up to 2 hours, there's a progressive relaxation of the DNA, a loss of excess supercoiling. And this is something that hasn't been detected previously in this system, at least not with the wild type enzyme. The wild type being the native form of the enzyme that is considered to be sort of the standard or reference protein sequence for the enzyme. And that's distinguished from mutant forms of the enzyme.
Steve, a question there.
Yes.
In this rightmost slide, each of those columns represent identical molecules, which
Yes, these are all the same plasmid.
They have the same knot type?
Okay, so these are predominantly, because of the fact that the enzyme produces mostly unlinked DNA, i.e. the unknot, you don't see the contribution here from most of the non-trivial knots. So in a sense, we're looking at the loss of supercoiling by the unknot, and we have to infer from that or use that as a proxy for the loss of supercoiling by the non-trivially knotted DNAs.
Thank you.
You cannot, the technical limitation here is you can't resolve on the same gel both knot type and superhelicity.
So Steve, the result, this one on the right is the result of processive recombination, more than one round of recombination?
Yes, there's definitely more than one round. Now, whether it's processive or not is something that I'm open to discussing with you, because in a sense, the reaction behaves quasi-processively. But I think it's based on the kinetics of the protein DNA interaction. So it's tied to the kinetics of the protein DNA interactions, not really a processive reaction in the same way that, for example, serine recombinases recombine DNA. And that's again something we'll come back to a bit later.
Okay, thanks.
Okay, so those are the gels. These are the quantitative analyses of those gels. This is the formation of knot types, various knot types as a function of time. And everything pretty much decays to an asymptote at around 30 minutes. So this graph doesn't go beyond 30 minutes, but you can see basically what happens. Interestingly enough, not all knot types are formed at the same rate, and nor do they decay in the same way. So the most probable knot type here, normal, by the way, this is the percent abundance of non-trivial knots that we have plotted here. So it's not the percent abundance of knots within the entire population. This is normalized to the population number of non-trivial knots only. And that's mainly because we can't easily quantitate the unknot band. It's just, it's so intense that when you try to quantitate the level of the unknot, there are huge uncertainties, simply because you've got small differences of large numbers. So we plot everything here in terms of the abundance of non-trivial knots, normalized in such a way that all of the non-trivial knots add up to 1. So the knot that forms first is basically the 5-1 knot. The 7-1, the 9-1, these decay rather slowly, but the 5-1 knot decays a little more rapidly, and at the same time that you can actually see the trefoil knot coming up. So interesting kinetics, not the kinetics I would have expected based on any reasonable model for recombination.
Steve, is this percentage of these knots, is that a function of the total length of the molecule?
It could be, yes. We did this only for this 4 kb plasmid. So you would expect more complex knots for the same superhelix density if you had a longer molecule, for sure. Yeah, so all of this really pertains to this particular length of DNA. At the same time, we follow the overall progress of the reaction and monitor the kinetics of formation and decay of different linking number topoisomers. And so each one of these has its own sort of characteristic kinetics. This is for a delta LK of -23, which is close to the mean value for natively supercoiled DNA, -22, -21, etc. And so we can quantitate that for each of those topoisomer bands. Okay, so that's more or less the experimental part of this project. Are there further questions?
Steve, could you go back to the just one slide, please?
To here?
Yeah. Okay. So would you be confident that beyond the 30, the slide before, just yeah.
This one here?
No, the next one, please. Just the one before questions. Okay. So would you be confident that beyond 30 minutes, these two kind of asymptotics of the 3151 and the 7191 would stay that way?
Yes, yes, yes. We've actually plotted, we have different versions of this graph. And yeah, there basically isn't much change after 30 minutes. Although, as you will see, some interesting things do tend to happen beyond 30 minutes. But they happen at a sufficiently low level that they don't affect these graphs, at least within the level of experimental uncertainty.
It's a very interesting thing, and I would have expect the 3-1 is more popular, and then eventually asymptote with the 5-1 rather than the other way around. Do you have any explanation for
I
this?
do, and I
also-
will. I do, and I will get to that in the second.
Okay, okay. Thank you, thank you.
Yes. One other thing I think I want to mention here is that I used to think that the reason why these experiments were generally done only over the course of maybe 30 minutes or so is because the enzyme itself could die, right? The enzyme, I thought that the enzyme probably loses activity after 30 minutes or so. But we did an experiment, which unfortunately I don't have the results to show you here, but if anyone's interested, I can share those results later, where we took an initial substrate plasmid, incubated with Cree, and over the course of, say, 2 hours. And then at various times, we would add to the reaction a separate plasmid, which is distinguishable from the first one using, for example, restriction digests. And so, what I expected in that control before we started this work was that you wouldn't see any Cree activity on the second substrate, the challenge substrate. But it turns out that Cree is fully active over 2 hours. So the asymptotes are not mainly due to loss of enzyme activity. They're due to something else. The enzyme remains, at least in our hands, fully active in the experiment out beyond 2 hours of incubation at 37 Celsius. Okay, so We decided to try and model this using a framework that we had developed a few years earlier to investigate type 2 topoisomerase activity and both knotting and supercoil relaxation in type 2 topoisomerases. And so I'm going to show you now some results of that modeling exercise, but I'd like you to take it all with a grain of salt. I love this quotation: all models are wrong, some models are useful. And I think we definitely conform to that maxim, which is actually due to a British mathematician by the name of George Box. Okay, so how do we approach this? Well, what we've done is to use techniques that we've used for many years now, based on Monte Carlo simulation, to model a quasi-static process on the DNA, where the enzyme action is slow relative to the equilibration of DNA conformations. And so essentially what we model here are thermal fluctuations using a Monte Carlo-based algorithm to sample configurations where the loxP sites are juxtaposed in a way that allows recombination to take place. And that basically means that they have to be within a certain radius of one another, and their orientations have to correspond roughly to the expected orientation of sites within a synaptic complex. And so these sites then just collide randomly. This is sometimes referred to as a random collision model, although I don't particularly like that term. But the essence of it is that the DNA can undergo many, many conformational fluctuations on the time scale that it takes for a recombination reaction to take place. If that were not the case enzymatically, I think you would end up with all kinds of sinks and sources, kinetically speaking, and you wouldn't get the kinds of results that we actually observe. You wouldn't get, for example, the distribution of knot types that we observe. So you can think of this as a Markov chain where you have a state A with a given delta LK and a given knot type that gets converted to a state B with a different delta LK and a different knot type. And the sampling of those transitions occurs through an equilibrium Monte Carlo simulation that uses a model we've used, as I said, for a long time, where you have a discrete worm-like chain that models the trajectory of the supercoiled and knotted DNA's duplex axis. So with this framework, you can now estimate the rate constant for a transition between these two states from some proportionality constant K0 times a factor that we call J, which is the equilibrium probability of juxtaposing the sites, multiplied by a transition probability that takes you from state A to state B. So this is the conditional probability of getting a topology B from a topology A, given this juxtaposition in state A. Any questions on this? Okay, so this is basically a snapshot from the simulation. This is a pair of juxtaposed loxP sites. And by the way, the loxP sites, unlike a type 2 topoisomerase situation where synapses can occur for any pair of sites along the molecule. In this model, we identify specific DNA segments as loxP segments in the model. So this is a juxtaposition, and this is the product, which is a trefoil. And now you can recombine that. Again, this shows the transaction that takes place during recombination. Okay, so what recombination will do is somewhat counterintuitively, it reduces the free energy of the system. Basically, what you're doing is you're taking supercoils and you're converting those into knot crossings. And so while you might intuitively think that either keeps the free energy of the system constant or perhaps even raises the free energy of the system, that's not the case, because you actually gain quite a lot of entropy by converting those supercoils into knot crossings. And so those knot crossings now can kind of re-equilibrate around the structure in more ways than supercoils can. And so this reduces the free energy of the system. But if the system equilibrated rapidly just via this recombination process, you would expect to get to thermal equilibrium relatively quickly, but maybe not fast enough to really fully relax the DNA. Moreover, you would expect after recombination that more complex knot types can form during a second round of recombination. So we're not restricted to only one round of recombination here. In fact, in order for this simulation to work and not violate principles like detailed balance, the way that the simulation is actually conducted, you carry out synapsis, you do a virtual pair of segment exchanges, as I showed you, and then you analyze the knot type that results, but you don't actually go down that pathway. You go back to your original conformation, allow the DNA to fluctuate some more, and then for the next synapsis event, you do the same thing. So you always stay within the original knot type of the molecule that's being simulated, and that's essential. Otherwise, the simulation wouldn't work. But we can take one of these torus knot products, which are all products of the first round of recombination, and subject those to a second round of recombination and ask what happens. And what happens if you allow additional rounds of recombination is that the knot type distribution basically explodes. So you get composite knots, 3-1, 3-1, 3-1, 5-1, 3-1, 7-1. You get 4-1 knots, you get 6-2, you get 8-2. And this is not observed experimentally. If you recall, the only knot types that were really present to any appreciable extent, except for maybe at very long times, these were all torus knots that you see above.
And so, Steve, this second round of recombination would be a random collision with a fixed knot type?
That's right. That would assume that it would be random collision with a fixed knot type.
Right.
Yes, that's exactly right. Yeah, we don't make any assumptions here about processivity.
Okay.
So once recombination takes place virtually, the molecule then goes back to its original knot type and just rejoins the ensemble, if you will, of knot type. So yes. So but you can take one of these 3-1, 5-1, 7-1 knots, which will still have residual supercoiling because we don't allow for supercoil relaxation to occur directly in the simulation. Again, we don't want to violate detailed balance. And then allow that structure to undergo a second round of recombination. And that's when you start to see this explosion of different knot types.
Steve,
Yes.
if I were to interpret dynamically the process upon recombination, would it be possible to say something, or would it be correct to say that after recombination there is a kind of fast, due to, I mean, related to the energy relaxation process, a fast kind of diffusion of twist along the
Yes,
along the...
well, it's not going to change the overall delta LK though. If
Sure.
if you have diffusion of twist, that doesn't really help you get out of this situation. You have to have, and in fact, that's what we postulate, there has to be some coordinated relaxation of torsional stress or supercoiling.
Thanks.
Yes. And that's what gets us out of this problem. So strictly speaking, this quasi-static random collision model can't be correct because of the relative absence of second round recombination products. And that's where we include some DNA supercoil relaxation activity. So now the notion is that you have recombination and torsional relaxation going on in the same experiment. And that's supported by our data because we do see relaxation of supercoiling. So this would allow for some thermal equilibration of delta LK for a given fixed knot type. And essentially diagrammatically, what we're looking at is something like this, where you start in the unknot state with high levels of supercoiling. The dominant pathway that we observe experimentally seems to give unlinked products. So that's this black dashed pathway here, which takes you from the unknot to the unknot via what is effectively equivalent to a topoisomerase activity, similar perhaps to a type 1 topoisomerase activity, although I don't think it's quite exactly that. But we do also see, of course, other knot types, 3-1, 5-1, 7-1, and so forth. And so these will also have to equilibrate somehow, although they don't quite get to the equilibrium unlinked state. And by the way, these free energies are actually free energies that we know how to calculate rigorously using methods that we've developed previously. So the mechanism, the natural mechanism of the enzyme has to involve some torsional relaxation to achieve unlinked states or less complex knotted states than would otherwise occur if you had simple quasi-static random collision. Now, mechanistically, how does this happen? Effectively, you have to have one of two things happen, I think. You either have to have some partial dissociation of the enzyme, which would allow for a local relaxation move in the molecule prior to the next round of recombination. So you can imagine perhaps that this synaptic complex here, which is shown by this ball structure, that this essentially divides into hemispheres, which can rotate freely relative to one another and then reassociate. That's one possibility. It's not a very satisfying one, but I think nevertheless it's a model we have to consider. One of the things that Zhizhang Lei, the PhD student who's leading this project, is working on now is trying to actually estimate the dissociation rates of the protein and see whether those would be consistent with such a model. If you have such a model, you can actually now look at what we call for those of you who are familiar with, for example, fluorescence spectroscopy, a Jablonski diagram for knot type, which is basically a diagram that shows the various pathways that take you from, in this case, the initial state, supercoiled unknot, to various other knotted states along the pathway. And one of the things I'd like you to notice here is that there is, in fact, a pathway that allows you to get to the trefoil knot of opposite sign handedness. So the negative trefoil versus the positive trefoil. And that is something that we actually observe experimentally, as I'll show you in a few slides.
Steve, can you go back?
Yeah.
Can you please go back?
Yep.
What are these circles which you drew there?
Yes. So these are various states of the different knot types.
So those are things
how
you
they
observe?
they decay to a new knot type, but with a different delta LK value. And so these are all of the pathways, well, actually, all of the pathways where the resulting product has an abundance of 5% or more, because there are some very minor products that we don't look at. But yeah, these are all of the different pathways that take you to the most abundant species via this network of states.
Thanks.
Okay. Now, one important point is if you invoke some torsional relaxation, we actually get a pretty good agreement, at least semi-quantitative agreement with the experiment. So this is the experimental kinetic plot that I showed you previously. And if you allow for torsional relaxation now, where the torsional strain or the supercoiling in the DNA chain decays as a function of time, you actually can get pretty good agreement with what we observe experimentally. So I think the model gets some traction here, based on what we've observed experimentally, and based on what I think is at least a reasonable model for how the enzyme has to work. In this experiment here, this is a different gel. All of these experiments were done at least in triplicate. And in one experiment, Zhizhang got the experimental conditions just right, where we could start to see the appearance of a second non-trivial knot band that appeared right below the main knot band. And we were able to assign these bands on the basis of mobility of knots of different handedness that were generated by using supercoiled DNA that had positive supercoiling instead of negative supercoiling. So in lane C over here, these are the normal torus knotted products from the first round or early rounds of recombination using negatively supercoiled DNA. But these are the products over here that come from positively supercoiled DNA. And you can see that, oddly enough, there's a difference in mobility. So that sort of implies that there's some chiral interaction between the knot of a given topological sign and the gel, which I think is worthy of further exploration, but probably not something we're going to do, although it's a useful method for trying to resolve these separate bands here. And probably they occur in the case of 3-1, but maybe weren't resolved on this particular gel. But you can certainly see this is the case for the 5-1 and the 7-1. And these are species that begin to appear at around 60 minutes and beyond. So that further supports the idea that these pathways allow you to go through some state which allows you now to change the handedness of the knotted product. Okay, so
Steve,
yeah.
just a quick point. So would you regard the pairs of chirality generated by an achiral knot parent?
Probably these have to go through some achiral knotted product at some point.
It's an
Yes.
interesting bifurcation.
Yes. Yes.
Dynamical system then.
Yes. Yes, there's definitely some symmetry here that I think is very interesting. Okay, so one alternative to the tetramer dissociation model that I spoke of earlier, where the synaptic complex basically splits into two and then allows for free relative rotation of the two halves of the complex before reassociating, is to have, and I don't think these models are necessarily mutually exclusive, but is to have a sign inversion take place during recombination. And so to DeWitt's earlier question, are the sites actually anti-parallel? We actually did a set of experiments that we published. Sorry. We actually did a set of experiments several years ago to try to determine what the geometry of the longest lived intermediate is in site synapses. And what Masashura did was to construct a set of loxP substrates that vary systematically in length. And she was able to determine from the phase relationship of the recombination reactions on those different substrates, she was actually able to determine that the synaptic complex doesn't have the structure that's seen in crystal structures of such complexes, but rather has a rotation of the two halves of the synaptic complex relative to each other by about 60 degrees. And so the sites seem, in our hands at least, kinetically, to form stable right-handed intermediate structures during recombination, which then could be resolved via some sort of planar intermediate, and then finally a a product of opposite handedness or opposite sign crossing during recombination. This is actually entirely consistent with a model from DeWitt and Nick's paper that I cited earlier, where they showed a model consistent with all of their data from all of the integrase superfamily recombinases that could form such structures during recombination and resolution of the intermediate form. Okay, well, I'm a little bit over time. Let me just summarize. DNA supercoiling plays a hidden role here in the distribution of Cree product topologies. And this is something that's not normally detected experimentally. Certainly DNA supercoiling does drive knot formation in Cree loxP recombination, which we can model as a set of kinetic pathways in a discrete linking number knot type space. And I think this is a more generally useful tool in DNA enzymology, and encourage others to look at these problems from that perspective. There's a biological motivation in nature for these systems to have supercoiling relaxation activity, because generating unlinked molecules is essential for DNA segregation to daughter cells, or to make new plasmid or rather phage genomes. And I'm still troubled by the fact that I'm not entirely comfortable with our physical model for relaxation activity. I think this remains an open question, but I'm certainly open to ideas about how this might occur. This is primarily the work of Zhizhang Lei, who's a third-year graduate student in my lab, along with participation of Rick Zeraldo, who was a postdoc, Ryan Compton, who was a technician. We got a lot of help from Eric Rawdon in knot characterization, and Andreas Hanke has been involved in a lot of the simulation work, both for this project and also for an earlier paper or set of papers that we published on topo-2 mechanisms. And this is my obligatory conflict of interest slide. I'm a co-founder and chief technical officer of Phenomics Inc., a company that I co-founded in the Bay Area here. This is a company that has technologies related to circular DNA, but has no interest, financial or otherwise, in connection with the work that I've presented here. And so if there's time for further questions, I'm happy to take them.
Okay, Steve, great talk. So questions? So I still didn't understand how you were able to compute delta LK when the knot type changed, or even when the knot type didn't change. I guess if they're all unknotted and you're sure about that, then you can look at the gel, but.
Right. So in the simulation, right. So actually, there's perhaps an even better picture that I can show. I have to go all the way back to the beginning, sorry. Well, okay, let's look at this image here.
Yeah, the Jablonski diagram. How do you
that's, yeah, we can talk about that next, but basically to your question, DeWitt, so we keep track of, we don't allow for any extraneous change here in supercoiling through the recombination process itself. So what we allow for is a synapsis event And then when recombination takes place, all of these trapped supercoils get converted into knot crossings. And so basically all of these nodes are conserved in terms of number of nodes, but the sign of 3 of those nodes in this particular instance change.
Right.
So we don't allow in the simulation for changes in delta LK. If we were to do that, that would violate detailed balance.
How do you observe it in an experiment, though?
Okay, in the experiment, what we have to do is we have to analyze the topoisomers of the products. So these are the distribution of topoisomers.
And they're all for that one.
These are the distributions of topoisomers that result from recombination. And you wouldn't see these, of course, with a plasmid that doesn't have loxPs. So there's no extraneous relaxation activity. It's all driven by Cree. But that is in fact a distribution that consists primarily of unknot DNA.
Right.
Because unknot DNA dominates the products. Right. So we're assuming, I guess if there's an assumption here, the assumption is that the level of relaxation that's taking place in the knotted products is the same as what's going on with the unknot.
Okay, yeah, that would be reasonable, yeah.
Steve, can you elaborate a little bit? You saw positive and negative torus knots. How do you...
At long times, yes.
So how do you envision that they come about, since you have negative
Right.
supercoils initially?
I think what happens is there has to be an equilibration of knot type thermodynamically, which is assisted by the relaxation activity. And you can see here from the pathway diagram that there are, in fact, pathways that will take you to knots of opposite handedness. For example, this 6-1, the 3-1 minus. And that's something we just observe in the simulation. So when we carried out the simulation, we also computed knot chirality, not just knot type. So we used a version of the Homefly polynomial that allowed us to get the chirality as well as the knot type itself.
I'm still a little puzzled. So can you elaborate how one particular pathway? So for example, if I see the 3-1 minus there, what is the prior stage of that? How do I read this diagram?
Right. So probably one pathway, for example, would be from say 7-1 to 4-1, and then from 4-1 to 3-1 minus, 3-1 plus or 3-1 minus. You could probably get both out of the 4-1. Now, we don't observe the 4-1 directly. I don't know if that's because it's kinetically less stable. We do observe some 6-noded knots, but those could also be composite knots too from round 2.
Okay, thanks.
Steve, I have a question, if I may. Christian here.
Of course. Hi.
I enjoyed very much the talk. The results are beautiful, both the experiments and the model. So I was wondering regarding experiments. I imagine that is not feasible, but I wonder if in perspective it would be. You have some beautiful gels, let's say for the superhelical density, uh, which is what you showed just a couple of slides ago. So I'm wondering, would you be able, let's say, to cut out some bands from that gel and then retrieve the DNA material and then nick it and then profile what is the knot density? I imagine you lose material as you go, no? But maybe that the information about knots is still there in those gels. So I'm wondering, in perspective, could be feasible.
Right. So I think what you're suggesting, Christian, is that some of those topoisomer bands correspond to non-trivially knotted molecules.
Right. Yeah.
Yes.
Mhm.
Yes. Certainly the experiment's doable. I'm not sure that gel electrophoresis would be the method we'd want to use, though. Probably we would need to use some kind of single molecule method, AFM, maybe even nanopore electrophoresis. Yeah, I think that would be a good project for a PhD student. Unfortunately, I don't think I'm going to have the manpower to do that. But if someone is interested, I'm certainly happy to collaborate. We can make material available or whatever's necessary.
Thanks.
I have also a question, Steve. Given the fact that you said that all macromolecules have the same length, would make sense to improve the model you talked about by weighting the torsion function with the curvature function, because as you relax towards that knot, curvature is going to decrease, and so the bending energy is playing a part there. I would expect that a correction with a decrease of curvature as well as torsion. So torsion first, and then followed by a decrease in curvature, total square curvature, would have an effect.
Well, all of that is really incorporated in the Monte Carlo model. So what we have is a Boltzmann ensemble of supercoiled and knotted DNA chains. That's what we're modeling. So in a sense, all of that is accounted for in the model, albeit not explicitly.
Oh, I see. Okay, thanks.
Okay, if there are no other questions, let's thank our speaker for a great talk.
Yes.